Good afternoon, everyone. Thank you for staying until the end of the day. And I'm a faculty member. I'm a professor of statistics and data science at UCLA. It is my great pleasure to present our recent work at this last session today to our audience. So this work is a very, I would say it's a combination of both research and tutorial. The reason is that we try to do the categorization of 31 existing computational methods for detecting spatially variable genes from spatial transcriptomics data. This is a very hot topic, as you can see from the number of methods. So I would say that our categorization is our research efforts. In particular, for method developers, which I assume to be many audiences here, we hope to provide you some insights behind these seemingly diverse methods. Also, it is like a tutorial because I'll talk about some of the basics shared by some of the methods. So this is a joint work with my PhD student Guang Ao and a former visiting undergraduate student Shuo, who's currently at Stanford. So this is my group website. If you want to check out this link to the preprint, you can find it under our publications. So first of all, I want to introduce the spatial transcriptomics technologies. As you have heard from Ben's talks before, so there are two major types of technologies, sequencing-based technologies and imaging-based technologies. They use different chemistry to measure gene expression levels in a tissue slice with spatial information recorded. And they have complementary advantages. For sequencing-based technologies, they can provide the whole transcriptome, which means that you can measure tens of thousands of genes for human. And what you lose is the spatial resolution. So for the most common technology called 10x Visium, developed by 10x Genomics, the spatial resolution is actually multicellular instead of single cellular. So you may have more than one, usually 10 to 20 cells on one spot. In contrast, the imaging-based technologies aim to detect RNA transcripts directly, but you have to design probes to target these transcripts so that you don't afford to measure the whole transcriptome. Instead, you typically measure 100 or 200 genes. So these are your targets, the genes of interest. But what you gain is the spatial resolution. You can even go to subcellular. So that's the reason both types of technologies are being used, and people will choose the technologies based on their research questions. So in spatial transcriptomics data analysis, there's a common concept called spatially variable genes, and this is in my title. So it's often short as SVG. And the definition of SVGs, what I want to trace it back to is this related concept for single-cell data analysis called highly variable genes, short as SVGs. I think this connection may be a good motivation of why we want to look into SVGs. So for both sets of genes, I regard them as informative features people want to screen for before they do further analysis. The fact is that even though we are interested in our whole transcriptome, but most of the genes may not be expressed or may not have high enough variance or variability to be interesting. So for single-cell analysis, HVG detection is usually one of the first steps. For example, in the most popular pipeline for single-cell data analysis called SURAT, you will usually screen for 2,000 genes out of 20,000 genes for human, and then you only look at those 2,000 genes afterwards. And you want to select the most highly variable genes, which means that the genes that have high expression variability across single cells. Because you assume that if a gene is functional, then the gene's expression should exhibit some changes. Otherwise, we don't understand why it is functional. So that's the assumption. And then people will rely on these genes for tasks such as cell clustering or identifying some even subpopulations for cells. So analogous to that, for spatial transcriptomics data analysis, SVG, to me, serve a similar purpose. But here, when you look at a gene's expression variation, you want to look across spatial locations instead of just single cells. So this figure shows the comparison. The left column is for single-cell data analysis, the right column for spatial data analysis. So you can see that for single cell, even though you can measure individual cells, but you lose their locations. You don't know which cells are close to which cells. But for the spatial transcriptomics data, you record the locations, even though the spots may not be single cell. They may be meta cells, but still you have the spatial information. Therefore, once you go to the second step for identifying these variable genes, for single cell, the only thing you can compute is a variance, one variance value per gene. But for spatially variable genes, you have the locations to consider. So therefore, you see that you need some additional considerations and techniques to detect these SVGs. And then what's next? So for single cell, after you get the highly variable genes, people typically rely on these genes to perform cell clustering, which can be potentially cell types. For spatial transcriptomics data, after you get the SVGs, people will similarly want to identify, for example, spatial domains, which are regions that exhibit similar gene expression levels, which Ben talked about this morning. But with the spatial information available, if you only calculate the variance for each gene without considering spatial locations, it may not be enough. For example, the left gene is a highly variable gene in the single cell sense. That is, you take one value per spot and you group the values to compute a variance. You don't care where the locations are. Then you can see that even though the gene has some sparse high expression levels there, here, and there, but they don't give you some informative pattern. Then this gene is a highly variable gene, but we don't want to call it a spatially variable gene. And the right gene is something people want to look for, because you can see that the spatial pattern where the gene is highly expressed is somewhat connected, right? It means there's a pattern here. So that is the very high level intuition behind the definition. So this is what we covered so far, 31 methods. Starting from 2018 until now, you see that probably 2022 is the busy year, right? Many methods were developed then. But as a method developer, I always think that we need to have a good motivation to develop a new method. Before we do that, we need to understand what's out there. What do these methods cover? What can they do? What can they not do? And also, we noticed something interesting. That is, these methods do not necessarily share the same definition. What is an SVG? If they don't have the same definition, it means that they're not comparable, right? Then benchmarking may not be fair, because if you use one method's definition as the standard for benchmarking, then you would definitely favor this method than other methods. So before our work, there is one review paper that covers 19 methods, and there were 3 benchmark studies which compared a different number of methods. However, these studies didn't focus on the categorization of SVG definitions. In other words, if you read the papers, whether these methods share the same definition of SVGs or have different definitions, it's unclear. So that's our motivation, and therefore our proposal is that by reading these papers, we propose 3 categories of SVGs. The overall SVGs are what I said in the beginning, the informative genes for downstream analysis. So you don't want to specify what they mean, but you just want to look for, say, 10% of all genes, like 2,000 out of 20,000, the genes that might be interesting and worth investigating. So therefore, the overall SVGs is the broadest category. And besides this, we have cell type specific SVGs and spatial domain marker SVGs. For cell type specific SVGs, people have cell type information about the spots, and they want to use this information. And when they say whether a gene is an SVG, it is within a cell type. So therefore, the cell type information is being conditional on. And to me, the reason of doing this is maybe we want to look for some interesting cell subpopulations or cell states within each cell type. And for spatial domain marker SVGs, this is related to Ben's talk in the morning, spatial domains. After you partition your tissue slice into spatial domains, you want to understand what is each domain, right? Then the idea or the straightforward idea is to look for the highly expressed genes in each domain and look for the gene's functions to assign the domain a name. So these are spatial domain marker SVGs. Then the next question is, with our categorization, what are the relationships among them? So I would say, theoretically, overall SVGs should include the latter two. But in reality, whether there is such an inclusion relationship would depend on specific methods, what each method assumes. So for this talk, especially the latter part, I will talk about the null and alternative hypothesis implemented in some typical methods. Then you can see that they are not exactly the same. All right, so following my categorization, here's an illustration. So suppose that we have a tissue slice as this, and each spot can be multiple cells. So suppose that we already know the cell types for the spots with some additional algorithms. And we have partitioned the spots into 3 domains. So if you were at Ben's talk this morning, there's one definition for spatial domains that is based on cell type proportions. So domain 1 has only cell type 1, domain 3 has only cell type 2, and domain 2 is an even mix of the 2 cell types. All right, then overall SVG detection or definition doesn't use cell type information or spatial domain information. So only the gene expression levels. So for example, this gene, which are highly expressed in this cluster of spots, may be regarded as an overall SVG. But for cell type specific SVG, as I said, we look within a cell type. Let's just look at cell type 2. Then within the spots that are in cell type 2, there is a gene that has some high expression in these spots. Then it can be cell type 2 specific SVG. Finally, for spatial domain marker SVGs, if we are interested in domain 2, what genes are highly expressed particularly in this domain, then this gene satisfies that. So therefore, you see that the latter 2 categories use more information than overall SVG. This one uses cell type, this one uses spatial domain. So with this categorization, now here's the new figure, right? So we colored the 31 methods based on their category. So green for overall SVG, which is the majority of the methods, and red for cell type specific SVGs, only 3 methods, and purple for spatial domain marker SVGs, 2 methods. So overall SVGs methods dominate the field. So now it's a hierarchy. This is something we drew to categorize these methods from a methodological point of view. So of course, we should do the categorization within each category, right? We should further divide overall SVG methods based on their methodologies. And because there are many fewer methods for the latter 2 categories, let's first look at them. For spatial domain marker SVGs, we look at the methods and we see whether they provide any statistical significance. This is what we call statistical inference, which means that for the detected SVGs, do we know how confident we are for them statistically, right? Like a p-value, for example. So for statistical inference, all of them, yes. And how do they do the inference? How do they assess the statistical significance of these genes? And both of them use regression model, and they model the effect as a fixed effect. So this is something I will cover in the last part of my talk, which is quite technical, about how do we assess the statistical significance. All right, so just bear with me that I will use the jargon for now before defining it. The third category, very similar, right? All 3 methods provide statistical inference, and they all use regression and fixed effect. Now let's look at this big category of overall SVGs. So for this category, the methods differ in many ways, but the first division is that whether they would reconstruct the data into a graph or not. If not, we would just use the 2D locations of the spots in the slice. We call them Euclidean space-based methods. If they connect the spots into a graph and then proceed from there, we call them graph-based methods. Now let's first look at the Euclidean space-based methods. And for these methods, we can further divide them based on whether they use some kernel or not. And later, I will show some example kernels so you can have better intuition. But for now, just take kernel as a jargon. So whether they use kernel or not, we can call them as kernel-free or kernel-based. For kernel-free methods, we see that 4 of them don't provide any statistical inference, which means that they will compute a statistic for each gene and then rank the genes based on the statistic. But that's it. They won't say where the statistical significance threshold should be. It's a ranking. For the rest that use statistical inference, they're very diverse. They can be categorized using Bayesian inference, or those don't use Bayesian inference. And for those that don't use Bayesian, I categorize them as regression fixed effect or dependence test. So for the last part, I said, we will give more discussion on these distinctions. But just for your information, there is a minority of methods that are Bayesian. And there are 2 named after Boost, and there will be a 3rd one named after Boost, and they were from the same author. All right, and for kernel-based methods, they use kernel, and they all provide statistical inference. I guess the reason is that if they want to use kernel, it's usually with a statistical model, then they can naturally provide some inference. And again, for the inference type, it can be Bayesian, you see Boost, and then regression random effect test, and dependence test. Especially for the regression random effect test, which also I will elaborate last, they can allow you to specify gene expression distribution in diverse ways. So some people said, oh, I will work with log transform counts, then I assume Gaussian. Some people said, no, I want to work with counts directly, then Poisson. Or if I want over dispersion, negative binomial, right? So there are many methods because of that. Finally, graph-based. So if we convert the spot-spot relationships into a graph first, then work with the graph, these are the methods. Again, some methods don't provide inference. For those that they do, they all belong to this type called dependence test, which means that they don't use a regression model. I guess the reason is that we don't have very good natural ways to do regression on a graph, usually. So that's why they use some statistical they construct some statistics and then perform tests based on that. All right, so this is the overview without going into details. But in the latter part, once I talk about the technical details, you will see them more clearly. So now it's the time for me to introduce some notations to formalize my discussion. So as all these methods look at one gene at a time. So for my discussion, I only need to focus on a given gene. And this gene has expression levels measured at n spots. All right, so I didn't know yi as the gene's expression level as spot I. So I is the index for spot. I will use this throughout. And just, as statisticians, we like to talk about random variables and the observed value. So to make some distinction, lowercase for observed value, uppercase for the random variable hypothesis. And 2D spatial location. Spot I, SI, because we are talking about 2D spatial transcriptomics data. It has 2 coordinates, SI1, SI2. Putting them together, I get the spatial location matrix, n by 2. Every row is 1 spot, the 2 columns for the 2 coordinates. And as I mentioned, some methods will use additional information such as spatial domains and cell types. So I call those inferred variables because they were usually inferred from the same spatial transcriptomics data. It's not like you know them beforehand, but you will use gene expression levels to infer them. So spatial domain is usually an indicator, like one spot is only in one domain. So therefore, we encode them into this vector. So suppose that there are a total of L domains, then spot I must be in one of the L domains. And we indicate that by 1, and the other values are 0. For cell types, suppose that we know there are K cell types. Then this spot I's cell type proportion should add up to 1. So for the spot level or the meta cell level data, a spot can correspond to multiple cell types. So therefore, you may have more than one entry, non-zero. But for the imaging-based technologies where the spots are super fine, then every spot has only one cell type, then this could be binary. So DI for spatial domain of spot I, CI for cell type of spot I. So from now on, I will focus on 2/3 of the methods that are using frequentist inference to detect SVGs. It's easier to just talk about them since they are the majority and they share the same statistical framework. So by frequentist inference, I don't know how many of you here are familiar with this jargon, but what it actually means, you can just simply interpret this as, oh, I will compute a p-value, because that's usually the end result of frequentist inference. So unlike Bayesian, you talk about posterior. In frequentist, you usually talk about p-value. So for frequentist inference, what it does is that for each gene, you would compute a test statistic. But this has to be your definition, and you can define it in diverse ways. And you want to know what is the distribution of this test statistic under your null hypothesis. Here, the null hypothesis would intuitively mean the gene is not an SVG. So under the null, under the uninteresting case, what is the value distribution for this test statistic? Then given a particular gene's statistic value and the null distribution, you would convert the test statistic to a p-value. So it's a 3-step procedure. So then I will talk about 2 major types of null hypothesis for these 21 methods. The first category is dependence test. So for these methods, the null hypothesis is independence. That is, the gene's expression level is independent of spatial location. For the second category, I call them regression-based tests. So first, you need a regression model. In the regression model, the gene's expression level is your response Y, and the spatial location is your covariate S. So what you want to ask, or the null hypothesis is the spatial location has no effect on the gene's expression level. And to implement this, we have 2 ways, fixed effect test and random effect test. Random effect test is also known as variance components test. So I think for our audience here who work on statistical genetics, this should be something you are familiar with. First, dependence test. So the null hypothesis is simply independence. So suppose that we have n spots, right, for one gene, then we have Y1S1 for spot 1. So this is the gene expression level, Y1. This is spot 1's 2-dimensional location, up to YNSM. And we assume that these are sampled from some joint distribution of the YNS, right, gene expression, spatial location. This is a null hypothesis. And for a particular method, if it rejects this null, then the gene is called an over SVG. For these 9 methods that use this formulation, we can further divide them based on the test statistics they use. So some of them use conventional test statistics, which means that their test statistics have theoretical null distribution. They can use directly. But some methods were very creative. They use unconventional test statistics, which do not have theoretical null distribution. Then what could they do? They use permutation. They use permutation to generate the empirical null distribution for their test statistic. And I will talk about 1 method per type. So conventional test statistic, SPARC-X. So if you know this field, SPARC is an earlier method, which I will also mention. And the same authors from Dr. Shang Zhou at Michigan, his group, they implement this fast version called SPARC-X, but it uses a completely different formulation. SPARC is regression-based. SPARC-X is dependence test. So SPARC-X basically compares 2 matrices, and each matrix is an n by n spot similarity matrix. So matrix 1 is just based on this gene's expression levels. They will define some similarity metric between 2 values, right, expression levels. Then they can get this n by n matrix. But matrix 2 will be based on spatial locations. So how can we model the n by n spatial location similarities? They use some kernel. Actually, they use 2 kernels, Gaussian kernel and cosine kernel, which are non-linear transformations, and then they can each capture a different pattern. So now let me jump my slides to show you the example. This is a Gaussian kernel. This is a 2-dimensional Gaussian kernel. This is the 3D visualization. This is the 2D visualization. So you can see in the Gaussian kernel, basically the pattern you are interested in is something like, they call it hotspot. So you are interested in like the middle is the highest center, and then as you get away from the center, you are more and more dissimilar. This is a Gaussian kernel. And they also look at cosine kernel. This is a cosine kernel. So cosine kernel will give you this periodic pattern. So they consider both kernels. Of course, each kernel has a tuning parameter, but they consider them both, and then they will aggregate the result. Without going into the detail, I will say that their test statistic is to compare these two matrices, one based on gene expression, one based on spatial location, and they use the Pearson correlation between the two matrices. And there is some theory to say that this Pearson correlation follows a mixture of chi-square distribution, and that's how they get the p-value. This is SparkX. Another method from the unconventional test statistic category is called single-cell haystack. This is quite interesting because you can see that this method applies some pre-processing to me, reduces the resolution of the data. For the gene expression, it performs binarization. That is, for a particular gene, it will divide its expression levels into just two states, detected and undetected, binarized. And for the spatial resolution, it will define use some predefined grid points to aggregate the spots into coarse spatial coordinates. Then they work with the binarized gene expression and the grid points. So to me, the idea is quite interesting because they use the distributional differences as their test statistic. So what do they do? First, they have a reference distribution based on all grid points without looking at the gene expression states. Second, given the gene expression state being detected or undetected, they can each have a conditional distribution of the grid points. Then their test statistic is the sum of two KL divergences. And one KL divergence is the divergence from the reference distribution to one conditional distribution. So you will get one KL divergence for each conditional distribution, and then you add up these two KL divergences. And in this case, for sure, we don't have a good theoretical null distribution. Then they do permutation. By permutation, what it means that they randomly shuffle the gene expression level across the spots and then compute their test statistic. All right, that's dependence test. So you can see that they are usually simpler in the way that they don't have a complicated model because they are just computing some test statistic they think as informative and then get a null distribution. Regression-based tests are the majority, especially the methods developed by people in statistics, biostatistics. So for this part, I will first do a review of this linear mixed effect model. Because I'm not sure how many of you have used this model before, so I think it will be a good idea to start with a review. So for linear mixed effect model, we have the linear model written in this way. Yi is the response. In our case, is a gene's expression level at spot I. We call it response variable. Beta 0 is an intercept. Xi is what we called fixed effect covariates of spot I. And beta is what we call fixed effects of those covariates. So we use just p-dimensional for the number of fixed effect covariates just as a general notation. And besides this term, right, the contribution of fixed effects, we have random effects. So Zi, which we call q-dimensional, are random effect covariates at spot I. Corresponding to them, their effects are called random effects, gamma, q-dimensional. And for this part, we typically assume the random effects to have mean 0 and some covariance matrix. And then we have the measurement error term, independent random error at spot I with mean 0. And typically, people would assume the random effect vector gamma and the random error vector epsilon to be independent. So for some of you who haven't learned this before, I guess your first question would be, why do we need to specify fixed effects and random effects? They seem arbitrary. Why? So I can share one practical reason, not a philosophical reason, but a practical reason. The practical reason is that usually your gamma if it is q-dimensional, you might have q to be large. That means you might have too many parameters to estimate if you want to estimate the gamma directly. So therefore, the compromise in the statistical way is that instead of estimating each component of gamma, we just want to estimate some joint distribution of the components of gamma. And in this case, our interest is shifted from gamma itself to the covariance matrix of gamma, or something we will simplify, right? So basically, the random effect model or mixed effect model can also be interpreted equivalently as a hierarchical model. That is, this is the bottom layer, but gamma itself, like gamma 1 to gamma q, this q-dimensional these Q components will jointly follow some hyperdistribution, and you are just interested in that hyperdistribution's parameter, which essentially reduces the number of parameters you need to estimate. Okay, so for the fixed effect, right? If we want to say, does my fixed effect covariance xi have any effect on yi? What we are looking at is the mean relationship. By mean relationship, what I mean is that if I look at the expectation of yi, the true value of the response, does it matter if I'm conditional on xi or not? If it doesn't matter, if the conditional expectation of yi given xi is the same as the expectation of yi, then it satisfies the null hypothesis. That is, xi makes no contribution to yi, and it's equivalent to beta being 0. So in fixed effect tests, you are basically testing the coefficient beta, whether it's 0 or not. For random effect tests, your focus is on the contribution of zi. But as we said, here, gamma is assumed to be random, right? So in this formulation, what is random? Yi is random, gamma is random, epsilon I is random. So the contribution of this term can be considered by this variance decomposition of yi. So we're using this rule called Tower rule to write the variance of yi into two terms. One is based on the contribution of zi, one is based on the contribution of epsilon I. So then if we say zi makes no contribution to yi's variance, then it's equivalent to testing that is equivalent to say the first term is 0. Then this translates to the null hypothesis being the covariance of gamma is 0. Okay, so you see random effects talk about variance, fixed effect tests talk about mean. And then we can generalize this linear mixed effect model to some non-Gaussian distributions. Because in this model, I said, if you write it in the form of distribution, if the error terms are normally distributed, and if you have given the mean yi being normally distributed, this is just another way to write the linear mixed effect model. Every distribution is Gaussian. And this is commonly used in statistical genetics. But what if yi is count, right? It's a count data, non-negative count. Then Gaussian doesn't seem to be reasonable. We can generalize by changing the distribution of yi from Gaussian to something else, such as Poisson. And once we change that, then for the relationship between the mean, distribution mean of yi, and the covariance, we also need to modify this part. And this is often called the link function, used in generalized linear model. But here we have the random effects, so it's called generalized linear mixed effect model. And we can even go to more complex property, like more complex models, such as what if the fixed effects, xi's effect is not linear, because xi transpose beta is linear, but we can make this nonlinear. So if we change xi transpose beta to some nonlinear function f, this is called non-parametric regression in statistics. So we can even have this generalized non-parametric fixed effect model, because you don't know the form, you may not know the form of f before, but you want to estimate it based on your data, right? You have to specify some basis functions and start from there to fit this model. So then the question is, for all those regression-based methods, they differ in their answers to this question. Should the spatial location Si be modeled as Xi, the fixed effect covariance, or Zi, the random effect covariance? That's the question. So I will first talk about the fixed effect tests methods. So there are 6 methods, and they are the only category that covers all 3 SVG definitions. So there's 1 method that detects overall SVGs, 3 methods that detect cell type specific SVGs, and 2 methods that detect spatial domain marker SVGs. So I will first talk about the SPADE method for overall SVG detection. So this method actually uses deep learning as its building tool. So what it does is that to convert spatial locations into fixed effect covariates, it uses a pre-trained convolutional neural network and apply it to the H&E image, which is usually accompanying the spatial transcriptomics data, like 10x Visium. You have spatial transcriptomics and you have an accompanying slide of H&E image. So it is applied, so this pre-trained CNN is applied to this H&E image to extract features and the number set to 512. These features will be used to represent the spatial locations in this linear model. So it's just a linear model. And basically, we have this vector XIS, 512 dimensional, and beta is the coefficient. So the test is just whether beta is 0 or not. If we reject this beta, we call the gene an overall SVG. So you see, the method on the testing part is very simple, and the challenge was thrown into this CNN for embedding the spatial locations. Okay. And for the test itself, the method uses lemma, which was a classical method developed for microarray data analysis using linear model. So you see, it's a combination of pre-trained CNN and lemma for the new purpose of detecting SVGs. SPVC is an exemplar method for detecting cell type specific SVGs. So you see that it's more complicated than the previous one because here the authors assume that the YIs are counts, the sequencing data counts. So then the distribution of YI given the mean is Poisson instead of Gaussian. And it has a 2-step procedure. The first step is that they use a reduced model without interactive effects between cell type, as spotlight CI, and spatial location SI. So you see in this model, in this relationship between the log transform mean, this is called the link function to the covariates. So here, the cell types is here, right? And the spatial location as a fixed effect is here. But there's no interaction. They are just add together. So in the first step, they will test whether both terms have non-negligible contributions, whether beta is 0, whether F0 is 0. If both null hypotheses are rejected, then it move on to the second step, which is a full model. In the full model, you see they added this additional term. So this Fk, which is a function of the spatial location Si, is based on cell type K. So it's within cell type K. So each cell type has its own function. So in this step, it's basically asking the question whether this additional term can explain the data better than the reduced model without the specific term, cell type specific term. So the second step is to test whether F1 to Fk are 0. If one Fk is not 0 or is rejected, then the gene is detected as an SVG specific to this cell type K. This is the second method, cell type specific fixed effect. Finally, spatial domain marker SVG. DE-space is a regression-based method for this task. So again, it works with counts, so yi given mu y is assumed to be negative binomial, and the negative binomial mean is assumed to be this function. So here, the domain enters here, right? So we use domains instead of the spatial locations SI. SI is gone, we use DI, and then we just want to test whether each domain has a non-zero contribution to the intercept. So if one of the betas is 0 and being rejected, then we have the gene detected as a marker for that domain. All right, random effect test. So for random effect test, the thing is that the spatial location will enter this random effect term. And there are 6 methods that use this formulation. Spatial DE is one of the first. So in this case, the spatial locations are encoded as a random effect covariate vector. So this vector's length is the same as the number of spots. If you have n spots, then Zi is n-dimensional. So how they code Zi? It's basically the dummy coding. That is, every spot has one in its own dimension. So like Zii is 1, and other Zij, if j doesn't equal to I, is 0. So you only have one in this vector. And you want to know whether the spatial location has any effect on Yi. So then gamma S is also n-dimensional. Every spot has its own coefficient or effect gamma I. But you have too many, right? Too many parameters to estimate. Then how do we use random effects to unify these n random effects into some common structure, common covariance, and only test if the covariance is 0? Then all these methods use kernel. How do they do it? So basically, they would formulate this covariance matrix of this n-dimensional gamma. This is an n by n matrix by assuming it to be some scalar multiplied to a kernel, a pre-specified kernel. And all these six methods detect over SVGs. So let me use spatial DE as one example. So essentially, here the covariance matrix is assumed to be a scalar, sigma S squared times a kernel matrix. And as I showed you before, the kernel can be Gaussian kernel, can be cosine kernel, or other kernels. Then the question for the null hypothesis is whether the scalar is 0 or not. So they want to test this null hypothesis. If they reject it, the gene is an overall SVG. All right, so now it's a time for discussion. So for the 26 methods out of the 31 that detect over SVGs, 9 of them uses kernel, 16 others don't use kernel. So then the natural question is, because kernel specifies the specific pattern you are interested in. So therefore, naturally, these 9 methods will have very good power if your interested genes follow these patterns, follow these targeted patterns. But they may miss some other genes that don't follow the pattern. So there's a power and specificity trade-off. This is something we can learn from the methodologies before doing any benchmark. And there are some remaining challenges which I see in the field. And these are related to whether your interested regions of interest are very small. Sometimes this could happen. For example, for cancer data, your interested region may be very small if you want to detect a spatial domain. So therefore, first, could you effectively identify it as a domain? Second, if you do identify it, could you easily identify its marker gene? So the first 2 issues are both related to the domain-related marker gene identification. In particular, for the second challenge, what I see is that all these methods don't talk about detecting patterns within a domain. If the domain is small, this will be more difficult. If the domain is large enough, maybe you can just apply the existing overall SVG methods to that domain. That's possible. And for the cell type specific SVGs, right, we only have 3 methods for that. What if the cell types you are interested in are small, right, small in proportion? And what if the cell types are not in your annotation? And also, all these methods to our knowledge, didn't assess their model goodness of fit. How do you know if your model is too complicated or doesn't fit well to data at all? Finally, there's another category of methods that don't call themselves SVG detection, but they're related. For example, B-layer is a method from Ben's group that helped to identify sharp changes in a tissue layer. So it's more looking for boundaries instead of the domains or the overall SVGs. And also, some method would want to use the H&E image as a helper, so to help us know the patterns we might be interested in. So what I see is that given we already have 31 methods, I think the next step is that we need more biological knowledge to guide us so we can say better what genes are interesting, what are not. Scalability is something we can definitely say for computational people, right? So for scalability, for most of the methods that compute a p-value, I would say there are 2 major steps that will determine the scalability. One is the time you spend on calculating the summary statistic. How long does it take? And the second step is how long does it take for you to calculate the p-value? For summary statistic calculation, spatial DE and Spark, they both use Gaussian process for their regression framework. And Gaussian process is known to be the order of n cubic. That is, if you have n spots, you need n cubic time to do the computation, which can be quite slow. So therefore, a method which was also in our summary, NNSVG, uses a new technique that speeds up the Gaussian process. It's called nearest neighbor Gaussian process, and it's an approximation. So this can reduce the computational complexity from n cubic to n, which is a big improvement. For p-value conversion, if the statistic has a theoretical null, like conventional statistics, it's not a problem. You can directly do the calculation. Otherwise, if it's unconventional, you need to add a time of permutation, which can take long. So therefore, for scalability improvement, especially if SVG detection is just the first step for screening genes, maybe I think the speed is very important. You don't want to have a too slow method. So approximation algorithms to speed up the summary statistic calculation is for sure important. And also some new strategies to reduce number of permutations is also very important. For future directions, first, I think existing methods, most of them don't focus on the differences between spatial transcriptomics technologies and biological differences. For example, imaging and sequencing-based technologies have different resolutions, and the spatial locations they measure have different random patterns, right? For some technologies, like the sequencing-based, the spots may be pre-registered in the grid, so they are structured. But for some imaging-based technologies, their spots are wherever the transcripts are expressed, so they're unstructured and random. For biological differences, we must consider whether the tissue structure is well-known, like brain, well-structured, or it's pretty messy, like tumor. And the current limitations is that most methods aim to be general. They can do everything. But to me, I think specifying technologies and tissue structure will likely make the discoveries more meaningful for interpretability. And also lack of consensus on pre-processing, whether we do log transformation or not, and one model to use, Poisson or negative binomial. And the second future direction, what I see is the possibility to enhance the statistical rigor and method benchmarking. So statistical rigor has been the focus in my group. In particular, this double dipping issue, what I see is actually widespread in both single cell and spatial data analysis. What it means is that you analyze the same data more than once, and there's a potential confirmation bias. For example, in the spatial domain marker detection, you use the same data to define domains, and then you use the data again to find markers. So same data twice. So what we have been doing in my group is this idea of using potential in silico negative control data to remove spurious discoveries. And I think having some fast visualization tools for our method developers is also very important, because we need something fast to help us quickly visualize the data. So I will just quickly say what we did for the negative control. This is built upon our simulator called SDesign3 for spatial data. And then this is a preprint. You can check it out. But this is to be updated because this is only for single cell. But now we have added spatial data. And the method is called cluster DE. Without going into detail, I'll just quickly say that cluster DE is a negative control contrastive approach. So the top row is your real data analysis. The bottom row is your in silico negative control data analysis. What we aim to do is that if we want to identify domain markers, we will make the synthetic negative control data to have no particular domains. So the gene expression change pattern is similar to real data, but there's no clear boundaries. Everything becomes smooth. Then if we use this negative control, being it reasonable or not, what we have? This is the result. So using this approach, you can see that the marker genes we detect, even we are using an existing pipeline, but just add a negative control, we can have more obvious marker genes compared to what you find. This is the result if you don't do the negative control. You do the standard spatial clustering or domain identification followed by DE. You may get genes like this. So their expression boundaries are not as clear as the ones we found. All right, so benchmarking, method benchmarking. What I see as a particular challenge is the lack of ground truth, right? What is true SVG? People may have different opinions, and we do not have many enough well-annotated data sets to do the benchmark. And so therefore, synthetic data sets can be a compromise or alternative, and there have been some simulators developed by us and other people. But the take-home message I want to say is that no method is optimal in every aspect. So therefore, method benchmarking should be specific to data characteristics like technology, biology, and align with biological questions. What do you want to do with the genes? Finally, I want to thank my PhD student Guang Ao for leading the efforts and Shuo for her great contribution to our literature review. And this is the preprint for the comment. I just want to say that we are revising the manuscript and it will become 33. So 31 to 33. All right, thank you very much.